everyone. We are proud to present the concept for the Microplastics Identification and Quantification System by Team MAPZ. That is Rosalind Matlow, Victor Azuma, Toni-Ann Peck, and Nyasha Zimunu. Our team wants to help solve the problem of microplastics pollution. In our research, we've determined that over 35 million tons of plastic are generated in the US each year, but only 8% of this plastic is recycled. 37% of plastic waste ends up in nature and undergoes mechanical degradation. We spoke to many different stakeholders from different walks of life, areas of study, and places in the world. And we received a lot of different suggestions for how to go forward with helping them to remove microplastics in the water and analyze the samples that they take. Our stakeholders suggested several different approaches to the problem of microplastic pollution. First and foremost, they said that the main problem we see is a heavy societal dependence on plastics. So anything that could reduce our dependence on plastics would help in this realm. Short of that, the most helpful approach to removing microplastics from the water is to remove macroplastics from water systems that would degrade into microplastics later on. It is also very beneficial to aid in the quantification of microplastic samples, as this process is very time-consuming and makes it difficult to publish information on microplastics pollution. Another large help in removing microplastics from our water systems is to reduce those entering waste management systems. Microplastics are often shed from nylon-based clothing and will flow out of washing machines when clothes are washed. Finally, if nothing else can be done, removing microplastics from the water systems is the only other way to prevent microplastics from accumulating. The needs expressed previously, as well as a couple of others, are listed here in more precise and concise terms.
With the interactions with our stakeholders, the 4 team members individually came up with 4 different solutions. One was focused more on the filtering section, and that was aimed at tackling the problem at the source. The picture that you're seeing over there is of a ball that could be added to washing machines to collect microfibers from the washing machine. And then the other project was looking more at counting the microplastics from samples, and this was geared towards helping researchers quantify and get an idea of the scope of the problem. And the third one was aimed and focused at removing microplastics in the ocean, and this was sort of adding a deep water functionality to an existing method. And then the last idea was looking at reusing some of these microplastics in 3D printers.
We compared the ideas we came up with to the needs that our stakeholders described. We created a poll to help in the down selection process, linking point values to each of the answers that we selected in our poll.
So to gauge which project we wanted to down select to, we had 5 different metrics that we were looking at, and these were all included in a poll, 5 different questions that we all had to answer, and those were looking at understanding the technologies, the interest, gauging our understanding of the skills required, the applicability to study area, and also personal choice. And this was to sort of widen our answers and sort of the scope that we were looking at these problems in order to down select. And using these metrics, we realized that like 78% of all points came across for MIQS. So we decided to work on the MIQS project, and it was the most favored comparing it to the other solutions that were proposed.
Here's a basic 2x2 for the niches of each of the ideas that we considered. At the top left, you see several items that are trying to combat plastic pollution at the macroplastic level. These are things like the 3D printing, filament, and plastic pyrolysis machines that recycle plastics at the macro scale. Moving farther to the right into the center, you end up with the washing machine filter that we described earlier, which is directed towards the removal of microplastics, but at their production before they actually enter streams and oceans. Moving farther to the right and remaining in the top half of the 2x2, we end up with the scaling of novel filters and the microplastics collection ROV. Both of these are directed towards collecting microplastics basically in the ocean or in free streams. However, the ROV is waiting until plastics are completely already in the environment, whereas the scaling of novel filters can possibly be implemented at the introduction of microplastics into streams, for instance, at waste management facilities. Finally, moving to the lower right quadrant, we find the Aquilus, which is a project from last year that was intended to help with the consistent sampling of microplastics, and our idea, which goes hand in hand with it, the microplastics quantification system. Both of these are meant to help researchers in consistently collecting data and quickly processing it so that papers can be produced and published that describe the possible dangers of microplastics in the environment. The main problems that we see with microplastics and trying to quantify and identify them is based on their size and the fact that they are often found in spaces with lots of biological contaminants. Microplastics are on the same scale as sediment and plankton and are often biofouled by those same size creatures. Many studies require plastics in samples to be counted by hand, so our device will be very helpful in trying to quantify microplastics. The chemical compositions of microplastics are very difficult to determine without spectroscopy, and even using this tool, because microplastics are often biofouled, it makes it difficult to identify the chemical composition of many of plastics in samples.
So my interpretation of this system is that it is going to include a chamber where we are going to host a capacitor. But before the microplastics could reach that chamber, we will mix them with a non-conductive fluid. The expectation is the microplastics that we're using have been cleaned and they're dry, so we're only leading microplastics into this chamber and system. So the microplastics will and the oil mixture would go pass through the chamber with the capacitor, where they will interact with the capacitor. Ideally being that with each interaction, we will see some sort of change in capacitance, and we'll be measuring this using a meter, like a voltmeter, and hopefully we'll also connect this to a screen, such as a laptop, for example, where the user will be able to view this, but we'll also include a counter as well, so that the number of microplastics could be counted via these interactions. And once the microplastics have interacted with the capacitor, they would be led down to a collection tank, where we'll be storing all the microplastics that have gone through the system.
My idea for MIQS is something like a small cylindrical receptacle with an opening for the glass petri dish that a scientist might put a microplastic sample in, already treated and ready to be reviewed. On top of this thing would be a camera and microprocessor unit that would be able to capture both images of this sample with visible light and on the IR spectrum using like a camera that has both visible and IR capabilities. And then the microprocessor on board would process these images into a set of, say, coordinates and images that describe how many microplastics are in the sample and how many of the items in the sample are some other kind of material. This would all hook up to someone's computer via USB or a similar universal cable, and a person could get that information using the serial bus.
My vision of how our system will look like is as follows. So we want to be able to filter off microplastics, make sure there are not any contaminants. And then once we have that sample, we want to be able to quantify the microplastics in it and identify them as well, which is, identifying them helps with quantifying. So for the microplastics are generally in the range of millimeters, even up to nanometers. So the smaller ones, we should be able to, capture the image or use a camera and our detection algorithm to determine which ones are microplastics and quantify them as well. So once they get smaller, that's when we will be using a microscope, which is shown in this diagram, and then whatever image is captured by the microscope is shown on the larger screen, and then our algorithm will analyze it. How do we plan to train our algorithm? So we plan to collect as many sample images of microplastics as possible. So one idea had been to buy microplastics, like plastic pellets, small ones, but we realized that that would be the first thing we would do if we actually didn't have access to the rivers. But the best thing we can do is use actual degraded plastic, which has been in the water, because the more time it spends in the water, the more the plastics chemically change. So we want to be as close to what microplastics look like as possible. And then after that, train our algorithm first. If we see that it can distinguish maybe two microplastics, count them accurately, keep scaling up as such.
So one way we could work on separating microplastics is to use, by putting the microplastics in the water, and then you put oil on it. You shake them so that the microplastics, which are more hydrophobic, have a higher affinity to join the oil. After we have that, we would basically place the microplastics in a funnel into a microfluidic channel, and because of the high surface tension that occurs at microfluidic levels, you would have the oil forming airtight bubbles that will trap the microplastics of comparable size with them. So what we have is that we end up seeing the oil bubbles in channels, and the ones that have microplastics in them would have different light refractive abilities, and that will allow us to account for the number of microplastics basically that have been captured via the number of light bubbles with different light refractive properties. Thank you. Another way we could separate microplastics is by using the dielectric properties of the microplastics. So in this case, what we have is that we are having microfluidic channels that are surrounded by capacitors. So you have the the water with the microplastics basically flowing through in bubbles, and bubbles that have the microplastics in them due to the presence of the plastic would have a very different dielectric constant compared to just free flowing water, and that will allow us to be to determine whether a microplastic has passed. Thank you.
This concludes our initial conceptualization for the microplastics identification and quantification system. Thank you so much for watching this video.